Hi everyone, welcome to my BioL 110 Ambassador Program video. I'm Wang Sheng. And today's topic actually came from me getting sick. Yes, I recently got a flu, the kind that knocks you out for days and makes you question all your life choices. But while lying in the bed feeling awful, I realized something. So we talk about virus all the time, but how many of us actually understand what a virus is? Why does it make you sick? Is it even live? And why does it spread so fast? So today I'm taking you on a quick journey into the world of viruses. We will look at what they are, how they infect us, and a small demonstration experiment I did, and why viruses matter in the real world. So let's go. Part 1. So what exactly is a virus? Imagine a biological USB stick. A virus is essentially a package, some genetic material, which can be DNA or RNA, wrapped in a protein shell called a capsid, and sometimes surrounded by a lipid envelope. That's it. Viruses have no cells, no metabolism, and cannot grow or reproduce on their own, floating on the surface or in the air. A virus is inert. It cannot do anything without a host. But once it gets inside a suitable host cell, it hijacks the cell's machinery to make copies of itself. Because of this dual nature, inert outside the cell, active inside, scientists often say viruses sit on the border between living and not living. Now, let's look at influenza as a concrete example, since that's what I caught. Influenza is an RNA virus, and its infection process is an elegant, if sinister, sequence of steps. First comes attachment. Proteins on the viral surface, most notably hemagglutinin or HA, bind to specific receptors on our respiratory epithelial cells. This is like a key fitting into a lock. Once attached, the virus enters the cell and releases its RNA, its instructional manual, into the cytoplasm. Now comes the hijacking stage. The viral RNA is copied, and viral proteins are produced using the host cell's ribosomes and enzymes. The host cell, which used to perform normal physiological tasks, is repurposed into a factory producing viral parts. Finally, assembled viruses are released. Influenza has another surface protein called neuraminidase, or Na, which helps newly formed viruses detach and spread to infect neighboring cells. Part 3. Why do we feel so awful when infected? Many of our symptoms, fever, sore throat, body aches, are caused not only by the virus, but by our immune response. Immune cells detect infected cells and release signaling molecules called cytokines. We trigger inflammation and recruit more immune cells to combat the infection. That inflammation helps clear the virus, but it also produces the symptoms we experience. Also, because viruses can replicate very quickly, the number of infectious particles can explode in a short time, which is why you can go from feeling fine to very sick within a day. In part 4, let's talk about a common real-life experiment, how COVID rapid tests work. Now let's take a break from symbiotic biology and look at something you've probably used many times during the pandemic, a COVID-19 antigen rapid test, also known as a lateral flow test. Even though it looks simple, the test is a brilliant piece of biochemistry. Here's how it works. Step 1. Sample collection. You swab your nose to collect viral particles. If they are present, these particles contain proteins called antigens from the SARS-CoV-2 virus. Step 2. Mixing with buffer solution. You dip the swab into a small tube of liquid buffer. This solution breaks open the virus and releases viral proteins. Step 3. The test strip. Inside the paper test strip is a microscope highway made of antibodies that recognize the COVID antigen, and gold nanoparticles attached to some of those antibodies. These gold particles make the lines turn visible when the antigen is detected. Step 4. Capillary action. When you add drops to the test device, the liquid flows along the strip, carrying your sample with it. Step 5. Detection. If viral antigens are present, they bind to the gold-labeled antibodies. This complex flows until it reaches the test line, where more antibodies capture it. The gold nanoparticles accumulate, forming a visible colored line. That's your positive result. And regardless of whether you are positive or negative, the control line must appear. It shows the liquid flowed correctly and the test worked. This test used the same fundamental ideas as laboratory immunoassays, antibody-antigen binding, but packs them into a cheap, portable 15-minute device. Technically, every time you take a COVID quick test, you are performing a simple immunology experiment at home. But why studying virus matters? Viruses shape our world in a profound ways. They can cause global pandemics, but they can also teach us about molecular biology, evolution, and even genetics. Some modern genetic tools, like CRISPR, were inspired by how microbes define themselves from viruses. So, understanding viruses helps us design vaccines, antiviral drugs, public health strategy, and diagnostic tools, like the rapid test we just talked about. And the more we learn about them, the better prepared we are for future outbreaks. So that's our journey today, from the influenza virus that recently knocked me out to the inner workings of viral infection, all the way to the everyday experiment sitting in your medicine cabinet, the COVID rapid test. Viruses might be tiny, but their impact is massive. And with a bit of science, we can understand them, fight them, and even use their tricks for good. Thanks for watching. If you enjoyed this video, please like, comment, and subscribe. And stay healthy right there.